As we age, we lose neurons. And in the retina, this presents, for instance, as the loss of retinal ganglion cells. Retinal ganglion cells are the cells that communicate visual information from the retina to the brain via the optic nerve. If a person develops glaucoma, superimposed on that natural loss of retinal ganglion cells that occurs during normal aging, we see an accelerated loss of retinal ganglion cells that can lead to visual impairment and blindness. Glaucoma is a highly age-dependent disease. The older a person gets, the higher the risk of glaucoma, as shown in this diagram. Here I show a schematic of the eye with the optic nerve pointing to the right. And if you take a histological section through the optic nerve, you'd see something like that. In green, these are the axons of the retinal ganglion cells on the way to the brain. In red, there are astrocytes, which are a type of supporting neurons, a type of supporting glial cells, which aid and support neurons and their axons. So our approach was to mine astrocytes for neuroprotective factors on the theory that astrocytes normally do protective work for neurons in the central nervous system and in the retina. So we took astrocytes of the optic nerve, did RNA sequencing, and looked at genes that are differentially expressed under different conditions, such as optic nerve damage and glaucoma. We then filtered the differentially expressed genes and looked especially for signaling molecules or transcription factors, and identified a few candidate molecules, and one of those I would like to talk about today. The protein in question is called SPP1, which just stands for secreted phosphoprotein 1. And under normal circumstances, as you see in this image of the optic nerve, it isn't really expressed. But if you inflict the damage to the optic nerve, which we did in this case by an optic nerve crush, the astrocytes become highly positive for SPP1. Now, that doesn't really prove that SPP1 is neuroprotective. So we did two lines of experiments, loss of function and gain of function experiments. In the loss of function experiments, we used a knockout mouse that is deficient in SPP1 and asked, is it more vulnerable to ganglion cell loss? So the first experiment we did was just to take the knockout mouse, put them on the shelf, and see how fast the ganglion cell loss proceeds. Shown in blue is the normal retinal ganglion cell loss that occurs during aging. 22 months is pretty old for a mouse. But in SPP1 knockout mice, here shown in red, the ganglion cell loss proceeds much faster. The next experiment we did is we used the glaucoma model and saw to see whether retinal ganglion cell loss proceeds faster in glaucoma too. Now, comparing the red and the blue bars in this graph, we see that yes, in glaucoma, of course, mice lose ganglion cells, but in the knockout mouse, they lose them faster. But mainly what really interests us is visual function. So we tested the pattern ERG, which is kind of an EEG just for the retina, and saw that in the knockout mouse, the electrical activity is more strongly diminished than in the wild-type mice, and the visual acuity also suffers more. The same is reflected in retinal axon counts. So if SPP1 knockout mice are more vulnerable, does overexpressing SPP1 actually help? For this, we used a virus, AAV2, that overexpresses SPP1 and also green fluorescent protein. We just used that to monitor and see what we're doing. So when we inject this AAV2 construct into the vitreous, we see pretty robust expression of SPP1 and EGFP in the retina, as shown in this picture. So we treated young mice with our AAV SPP1 construct, and again waited for a year. During this year, mice normally lose ganglion cells. However, if we pre-treat them with the AAV2 SPP1 construct, the ganglion cell loss they lose much less ganglion cells than the untreated mice. And even more interesting for us, they retain the electrical activity and the visual acuity in the retina better than the untreated mice. We can also see that in the EM. If you look at the middle picture, this is a rather old mouse, and you see one of these ganglion cell axons, which is really distended and swollen, and this is a degenerating axon. If treated with AAV2 SPP1, the optic nerve also morphologically looks better than the normally aged mice. So next we tried our AAV2 SPP1 construct in a mouse model of glaucoma, and again did the same kind of experiment. We treated the mice with the virus, then increased the intraocular pressure to inflict glaucoma, and then tested whether the ganglion cell numbers are, whether the mice preserve more ganglion cells, and especially whether they preserve visual acuity. And in both cases, in this case comparing the gray and the blue bars, the mouse preserves visual function much better, almost but not quite to normal levels. So we next tried whether you have to do that with a virus or whether the SPP1 protein itself would do. So in this case, we used a long-release form of SPP1 protein, injected it into the vitreous, and tested whether that would afford protection in a glaucoma model. And again, it does. It doesn't work quite as well as the virus, but it protects retinal ganglion cells, electrical activity of the ganglion cells, and visual acuity. So in a model of aging and in a model of glaucoma, SPP1 is highly neuroprotective. So our next steps would be that we want to use a screen for small molecules, because either a gene therapy, either be it AAV2 or a protein therapy, would have to be injected into the eye, and we are hoping to find small molecules that increase the expression of SPP1, and that could be given perhaps as an eye drop. There are some other conditions where we lose ganglion cells, traumatic optic neuropathy, for instance, or secondary glaucoma that can result from injury, or NION, which are more acute and maybe therefore easier to test. And finally, though we have not seen any negative side effects of overexpressing SPP1, we did so in a mouse. Mice don't live very long. So a glaucoma therapy, however, would have to be given for years, if not decades. So we are also interested in testing this treatment in a long-lived species, particularly in non-human primates.